Assistant Director in the Office of Technology Development at the Medical College of Wisconsin, where we celebrated our 125th anniversary last month. I'm so proud also to say, thanks to Dr. Cowley, that precision medicine and gene editing are alive and well at MCW. In fact, many of you may remember that in 2009, MCW became the first in the world to treat a rare disease through whole genome sequencing. Some of you may remember young Nick Volker's fascinating story called 1 in a Billion. I've also been a proud BioForward board member since 2013. I want to thank you as a board member, each one of you, for being here at this incredible summit. Are we inspired yet? Yeah. Are we having fun yet? Yeah. Are we making connections? Great. Let's give it up for Lisa, Brian, Mike, Kate, Jody, Nassim, and all of our volunteers, please. Thank you. Well, today I have the pleasure of introducing this very timely panel discussion, especially after Chris Mason's incredible keynote address this morning. What's next? Exploring the impact of gene editing on medicine, research, and the next generation, especially when we're already exploring clinical trials in human beings to explore how gene editing can help provide better treatments for very serious diseases. I'd like to thank our panelists. We're very excited to hear more from them, and I will introduce them very simply because you will find their bios in your programs. Our moderator will be Tom Foti at Aldevron. He'll be joined by Dominique Brossard at UW-Madison, Jill Wildonger, also at UW-Madison, and last but not least, Aron Geurts, who we fondly like to call the king of gene editing at MCW. Thank you.
Hi, everyone. So here we are to talk about CRISPR, a revolutionary technology, and as you can see in my first slide, I'm highlighting how CRISPR is really everywhere in the world around us. So how many of you have seen magazines that have CRISPR in the headlines, or seen an article about it in the newspaper? So hopefully many of you are raising your hand. So as you can see here, CRISPR has really gotten a lot of attention in the press, and it's gotten a lot of attention because it's really a revolutionary technique for us to edit the genomes of many different organisms. And while there are many applications for this genome engineering technology, the reason why we're here today, or one of the main reasons why we're here today, is to talk about how this technology could be applied to editing the human genome. So many of you, when I talk about genome editing and humans in the same sentence, are probably thinking about designer babies. And so how many of you, the first thought, conscious or subconscious, was designer babies? So some of you that probably just popped into your head. And one thing that I like to point out is that while we talk about designing babies to have traits that we like, I want to remind you that we all like different things in different people. And so one thing that I say is that I love my husband very much, but you may not find my husband to be your epitome of what a human being should be. So, and I say that with a lot of love. So we have, for example, here on this baby, we have things that we want for our children. We have things that we want to see in other people. And some of those things we have to learn, for example, kindness and traits like this. Other traits are more directly linked to genes, and we'll talk about that in the scenarios that we go through after this brief introduction on the CRISPR editing technology. So CRISPR is one new technology to edit the genome, but genome engineering has been around for a while, and there are some common goals for genome engineering. So one of the most basic goals for genome engineering is to understand how genes, RNAs, proteins function within cells. As genomic engineers, we also want to utilize these technologies for discovery approaches through genetic and chemical screens, to be able to apply this to model and treat human diseases, to, for example, create crops with desirable traits, to generate new fuel sources. And we can also use this technology to regulate disease vectors, for example, mosquitoes. So there are a lot of different ways in which we can apply genome editing to issues that we think about impacting us. So how do you engineer a genome? How do you edit a genome? So the way that you do it is by first breaking the genome and then letting the cell fix that break. So when the cell fixes this break in the genome, and I'm showing the genome as this bracelet, it can either fix it and make it just like the original, but the cell might also fix this break, and the outcome might be different than the original sequence. And so this is where this is what they're using to edit the genome. So changes to the genome can be introduced when the broken DNA is repaired, and genome engineering relies on breaking the DNA at a specific location and then changing that DNA sequence during the repair process. So in order to break the genome at a specific location, we use what are called nucleases, which are essentially DNA scissors. And so these DNA scissors are used to cut the genome at a specific place. In the CRISPR system, this DNA scissor is called Cas9. Now, once the DNA is broken, the cell's own DNA repair machinery will kick in to repair that break. And there are two key pathways to repair that break in the DNA, one of which is imprecise and one of which is precise. I want to emphasize that it's the cell that's determining how that broken DNA is repaired. So researchers are not able to dictate which repair pathway the cell will use. We can influence it, so sometimes we can bias the decision to make the cell try to pick one repair pathway over the other, but currently we can't dictate whether the cell will use an imprecise repair mechanism or a precise repair mechanism. And so it's definitely one thing to think about when we think about editing the human genome. So the researcher can determine where the genome is broken, but the cell will really take over to determine how that break is repaired. So the cell is in the driver's seat when we think about repairing that break to the DNA. So why has CRISPR been so revolutionary? So CRISPR has been so revolutionary because it's a very simple system to use. It's made of two parts, an RNA molecule that's called the guide molecule. It guides the nuclease, these DNA scissors, to a specific sequence in the genome. And then that nuclease, this DNA scissor, will cut the genome at that specific location. So when we think about cutting DNA, you can imagine that you want that cut to be very precise. I have a 4-year-old, and I would not trust my 4-year-old to walk around with a pair of scissors chopping stuff up. So we want, let's say, an adult in charge of the scissors. So we want the system to act like an adult with a pair of scissors. And so CRISPR is very specific. It has, while there are some variations in reports of off-target cleavage, it's actually fairly specific, and the frequency is likely to be quite low in most cases. So this is in contrast to some other genome engineering techniques that existed before CRISPR. These include zinc fingers and talons. These tools were quite difficult to create, and they also have been reported to be less specific and also work less efficiently. So where did CRISPR come from? And I really want to emphasize the origin of CRISPR to make it clear that we really need to continue to support and fund fundamental research. So CRISPR is the bacterial immune system, and it protects bacteria from viruses. And so one product that's really dependent upon healthy bacteria is yogurt. And so when we think about producing yogurt, we're thinking about keeping the bacteria healthy. And so there's been a lot of research on keeping bacteria healthy and how the bacteria protect themselves from viruses. And it turns out that CRISPR is the way that bacteria protect themselves from viruses. So if we think about the timelines of when CRISPR was first identified in bacteria, and when we first started to do genome engineering. So down here at the bottom, this is the timeline for genome engineering. So the initial reports on genome engineering were from the mid-1980s, and this actually dovetails when CRISPR was first described, and you can see that they proceeded along parallel pathways until about 5 years ago, when the gene editing potential of CRISPR was realized. And then these two pathways converged, and now CRISPR-based genome editing has really taken off. So let's come back to thinking about how CRISPR is applied. So we've talked about some of these before. So it's been used, for example, in crops and animals. It's used in synthetic biology. It's been used to study evolution. We use it in my lab to do fundamental biology. It's also been used to look at biofuels. So there are many different applications of the CRISPR technology. But I want to come back to this application that most people think of when we talk about genome engineering, which is how it might be applied to humans. And when we think about editing a human genome, there are some additional things that I think we should consider. So first of all, I want to remind you that you're more than just the genes that you got from your parents. So many of you probably know identical twins, and you know that these identical twins are not actually identical. They have different personalities, they have different traits. So you can start off with the same genomes, but end up with two very different people. And so we need to think about this interaction then between lifestyle, where you live, and your genome. And then the other thing to point out is that every cell in our body is constantly changing its genome. So as Carl Zimmer stated in May 2018, every cell in your body has the same DNA, except it doesn't. So we are exposed to many different mutagens during our lifetime, and those mutagens can change the genomes in different parts of our body. And I also want to point out that this is relevant when we think about some human diseases. So I'll talk about cancer. So you can be born with a predisposition to cancer. So for example, one copy of a gene that would cause cancer could be mutated and inherited from one of your parents. And then you can acquire a second hit during your lifetime and thus develop cancer. So I just want to end then with pointing out that there are a number of different things that we have to consider when we think about editing cells. Different cells can have different genomes, and then also there's more to who we are than just our genomes. And so with that, I'll say that I really look forward to talking with you about the different scenarios and hearing what you have to say about genome editing. Thanks very much.
Thank you, Jill, for that overview. Good afternoon, everyone. I also wanted to mention a quick thank you to Chris this morning for that outstanding talk and for Illumina making that happen. It was great to hear that. And at the end of his talk, he talked a little bit about genome editing. So looking forward to carrying on that conversation. I'm very humbled to be here with you today and engage this panel and the audience on this important topic of gene editing. Thank you to the BioForward team, Lisa Johnson, Brian Muller, Mike Harrison for this opportunity and putting this important topic on the agenda. I'm also honored to be on the stage today with our esteemed panelists, Aron, Jill, and Dominique. My name is Tom Foti. I'm Vice President and General Manager at Aldevron. Just one slide about Aldevron to give you some context of how we're interacting with the global CRISPR world. As you can see by the icons listed above, we have four scientific platforms: plasmid DNA, messenger RNA synthesis, which are based at our site in Fargo, North Dakota, protein manufacturing, and Cas9 manufacturing, which is based here in Madison, Wisconsin, and antibody development services, which are based in Freiburg, Germany. To date, we have manufactured greater than 50 lots of CRISPR nucleases for clients around the world. Aldevron differentiates with our ability to scale in batch size from small scale to large scale and from preclinical to clinical manufacturing. And the reason that's important is because Cas9 potentially has therapeutic benefits for clinical trials, and our ability to make Cas9 at a GMP quality grade is important to advance the field. We've been in business for 20 years, building our process development and manufacturing and quality systems, listening to our clients, and it really has fueled our growth. So that's a little bit about Aldevron. I do want to mention that our growth has fueled several job openings, and at the career fair, Samantha, shameless plug, Samantha will be at our career fair. So a little bit about NIH funding. In this slide, I wanted to highlight the growth in NIH funding as it relates to CRISPR research. No matter what metric you choose, you'll find explosive growth around CRISPR gene editing. As you can see in this chart, in 2012, there was $8 million of NIH funding for CRISPR-related activities. In 2017, this has increased to $884 million, and 2x growth over 2016. In 2018, we're expecting continued growth at the same pace. Moving to the next slide, it is not surprising that NIH funding has led to a significant increase in CRISPR-related publications. Similar to many other transformative technologies like PCR and sequencing, basic research funding is essential to translate this technology to useful products and new therapies for patients. Some of these NIH dollars most certainly have funded CRISPR-related work in the state of Wisconsin. Here you can see in the graph that the NIH funding has resulted in a sharp increase in publications, and in 2017, you can see that there's over 3,000 peer-reviewed journals written about CRISPR technology. Similar to the NIH funding, the investment capital has also flowed into many companies in recent years. In 2015, there were only 4 CRISPR-related companies. Today, there are 28, and several other of those companies are in stealth mode, so we don't know about them yet. The 3 largest CRISPR companies are Intellia, Editas, and CRISPR Therapeutics, and as you can see in the table above, they have gotten significant funding requirements. They have significant funding levels. Since 2014, over $2 billion has been poured into CRISPR-related companies. CRISPR Therapeutics recently announced a clinical trial that will be conducted in Germany, which hopes to cure patients suffering from beta thalassemia. Last week, Aldevron hosted a gene editing panel in San Diego, which I attended. Editas announced there that they are filing their first IND in October. So these companies have a lot going on, and they're moving to the clinical stage of their development quickly, and they're doing that with the help of large pharma. Intellia has partnered with Novartis, Editas has partnered with Celgene and Allergan, and CRISPR Therapeutics has joined forces with Bayer and Vertex. The investment in these companies has resulted in many preclinical and clinical stage therapeutic programs. As you can see in the graph, in 2014, there was only 6 preclinical programs. Today, there are 68. I wanted to mention that in 2018, there are 13 worldwide clinical trials, as indicated in the chart. 11 of them are being conducted in China. Due to the growth in gene editing clinical trials, there are many ongoing efforts between universities, agencies, and various countries to set guidelines to best use this technology. Today, we are very interested in this forum to get your feedback. And we're going to have an interactive polling tool that we're going to use to get your feedback. So please engage and be a part of the future of CRISPR. A little bit about what's going on in Wisconsin. The previous slide provided an overview of the market growth and the funding levels of CRISPR. Now I wanted to share a few things that are happening here. The Aldevron team in Madison has developed a large-scale GMP method to produce and QC test for gene editing enzymes. These are listed above. Aldevron is also the first company in the world to produce GMP Cas9 enzymes as a standard inventory product. This new product launch has enabled us to build collaborations with many of the leading gene editing companies in the world. Across the state, many Wisconsin institutions are very active in research around gene editing technologies. At the Medical College of Wisconsin, researchers are using gene editing techniques to investigate and model human disease in zebrafish, mice, and rats, focusing on diseases like hypertension, kidney disease, and diabetes. At the University of Madison and the Wisconsin Institute of Discovery, researchers are using gene editing tools to model human disease, create nanomedicines for the eye, engineer fruit flies to study the nervous system, and develop the next generation CAR-T therapies. Bioethics and life science communication experts from the University of Wisconsin-Madison are also very active in the ethical debates on gene editing, which include national and international committees that will be determining gene editing policy in the future. Marquette researchers are using gene-edited neurons to screen the factors that affect cell growth. University of Wisconsin-Milwaukee researchers are using gene-edited zebrafish to study their growth and survival. The Blood Center of Wisconsin uses gene editing to study vascular disease and find ways to combat different types of bleeding disorders. So you can see there's a lot going on in the state and in this very explosive growing field. So where have we been and where do we go from here? Looking at the timeline in the slide, I've highlighted a couple of milestones over the rapidly evolving last 6 years. In 2012, Jennifer Doudna and Emmanuel Charpentier published their first groundbreaking paper. That was just 6 years ago. In 2015, the National Academies of Science and Engineering and Medicine launched a human genome initiative to facilitate decision-making for the responsible use of gene editing research. In February of 2017, that committee published their first report titled Human Genome Editing, Science, Ethics, and Governance. The report identified 7 overarching principles to guide research and clinical use of human genome technologies. The committee concluded that in the report that gene editing trials should only be limited to the treatment and prevention of disease and disability. In 2018, as I mentioned, there are 2 clinical trials that have been initiated by CRISPR Therapeutics and the University of Pennsylvania. So with the background from Jill and this background on the growth in the market and the funding and the different companies, we would like to move to our next slide. So during this panel, we want to make sure we address the most burning questions that you have. Therefore, we are using this simple audience interaction platform called Slido. Slido allows you to submit your questions as well as upvoting questions for other participants. Be sure to upvote the questions you like. We also want to know your opinions on ethical questions and scenarios by voting using this live tool. So I was at CRISPRCon, which was a conference at MIT in June this past year, and in 2017 I was at Berkeley at the CRISPRCon event. They used this tool to facilitate and really get the audience involved. So I'm hoping that you will engage in this. A little bit about how to do it. So if everyone can grab their smartphones, and I'm serious about this, grab your smartphones. If you go to www.slido.com, if you enter the event code #Gene, we're going to do a 1-question practice, and then we're going to go through some live scenarios, which will help facilitate our discussions as we move through the rest of the program. One comment before we get into that, just about human genome editing as it relates to human germline editing. Human germline editing involves making changes to the genome that are passed on to future generations. That's the main difference with human germline editing versus editing somatic cells. The code is #gene. Oh, it's just gene? Okay. I'm just reading the slide. So, moving to Slido here, what should happen here, and you can see the live feedback that'll happen. So what best describes how you feel about the future of genome editing? Both hopeful and concerned, enthusiastic, not sure, pessimistic. So it's very interesting. I don't know what this, the data set is here. If there's, 300 people, that's very similar to what I saw at CRISPRCon in Boston earlier this year. So anyways, this is how the program's going to work with Slido. We're going to go through 2 different scenarios, and then we're going to ask a question about that different scenario, and then we're going to go to live questions for that particular scenario. And so we have 2 programs, or 2 parts of the program, and then we'll go through that process. Ask you for your feedback, ask you for questions. So, scenario 1. Cystic fibrosis is a genetic disease caused by one mutated gene from each parent passed to their offspring. The disorder causes serious damage to the lungs and digestive tract and is life-threatening. If we could safely utilize gene editing techniques to fix the mutated gene, it could spare individual families from suffering and may lead to a reduction in incidence of debilitating disease affecting 30,000 people alone in the US. Imagine you and your partner both carry the CF mutation. Your child would have 1 in 4 chance of being affected by cystic fibrosis. So give people an opportunity to respond to that, and then I'm going to sit down with our panelists and introduce them, and then they're going to respond to these questions and also to any other questions you might have. So go ahead and vote. While we're doing that, it's probably a good opportunity to introduce the panel. So Jill, maybe we'll start with you and give a little bit about your background and how you're involved in CRISPR, and we'll take it from there.
Sure. I'm Jill Wildonger. I'm an assistant professor in biochemistry at UW-Madison. And I got into CRISPR because I study how neurons develop, and we wanted to manipulate proteins and RNAs within the neurons themselves without having to add them exogenously. And this led us then naturally to genome engineering technology. So we work with fruit flies as a model system, and my lab was one of the first labs to adapt CRISPR for use in Drosophila. And we did so within 5 months of the technology being published. So it's a really fast-moving field, and that gives you a sense, too, of how easy it is to use this technology.
Dominique?
Hi, everyone. My name is Dominique Brossard. I'm a professor and chair in the Department of Life Sciences Communication at the University of Wisconsin-Madison in the College of Agricultural Life Sciences, such as Jill. I broadly construe study public opinion dynamics related to new technologies and particularly controversial technologies as they come to be. In another life, I was actually a life scientist, a geneticist, most particularly studying genetic engineering as applied to crops. And then I switched to the evil social sciences to study, what scientists do. But that dual hat actually gave me an entry point in a lot of work, particularly National Academy of Science. I was part of their report looking at genetic engineering crops, promises and futures. We were supposed to settle the debate. We just continue the debate. And more recently, studying public attitudes toward CRISPR-Cas9 and potential human gene editing, my department has been involved in the report that Tom was mentioning, as far as the human genome, ethical considerations. So we did the first large-scale survey of the population of United States to see how Americans felt about the technology, and I can also share those results with you later on. So delighted to be here with this nice panel.
Thank you.
Hi, my name is Aron Geurts. I'm from the Medical College of Wisconsin, and my laboratory uses technologies like CRISPR and zinc finger nuclease and TALENs that Jill presented to you all to engineer genomes to model a human disease in either animal models or human stem cells. And so one of the limiting factors that we often face when trying to understand how a disease mechanisms work is having a useful model in which to do the mechanistic studies and or test a potential therapy. And so for a lot of our work, we work with the rat model. You heard my chairman, Dr. Cowley, talk at lunch about cardiovascular and kidney disease that he studies, and he's one of my closest collaborators. And so we leverage the genomic research and the gene editing to try and create better models, more useful models to understand diseases like hypertension.
Thank you. So, are you guys voting on the? Oh, there we go.
Yes, for both.
It's a little bit. I'm gonna have to stand up over here so we can see this a little bit more. So 61% would. 61 said yes to both. 20 yes for me, but not for widespread correction. I don't know. Not for me, but yes for the public health. Kind of interesting. One of the things that I want to pass off to Aron as it relates to cystic fibrosis and as it relates to this topic of gene editing is, can you explain to the group here a little bit about the difference between germline editing and somatic cell editing?
Sure. So when we think about editing a germline, right? So we're talking about having an individual who has who carries a carrier of a gene mutation, perhaps, that could be transmitted to their offspring. And so when we think about germline editing, we're really talking about, can we use genome editing to prevent the propagation of that mutation onto the offspring? So I think everyone can imagine how engineering a germline would lead then, done successfully and safely, would lead to children who then would not carry the gene mutation. The other side is what we call somatic gene editing. So now you have someone who was already born with the gene mutation, or born with 2 copies of the gene mutation in the case of cystic fibrosis. And we're trying to use gene editing in the somatic cells, so the other tissues in the body that are not the germline, to try and correct that gene mutation or reduce the disease properties of that gene mutation in order to either extend the life or improve the patient's health. And so those are really the differences between the two.
Yeah. So thanks for that, Aron. Dominique, I was thinking, in your business of life science communication, and it's great to hear that you're on this National Academy of Medicine, helping to give feedback as relates to how we communicate to the world what's going on with CRISPR. Can you give us some examples or kind of maybe insights on the report that was published in 2017?
So the thing to keep in mind is that so the work we do in communication is more to understand the information processing going on. We don't particularly give advice on how to communicate to develop the technology. To some extent, I would say the role that we take is to represent the vast majority of people that don't say anything, and to have a way to integrate public voices in the debate. And I would say this is one of the take-home messages of that report, that there was a strong call for what we call public governance. That idea that for the new type of technology that now, let's say, scientific expertise led us to develop, very often the questions that are asked are not only questions that are scientific, but they raise social, legal, ethical implications that science alone cannot answer. And that's why we have panels such as these ones, where we can discuss, all of us, what we think. And so one of the take-home messages was that we shouldn't take for granted that experts know how to move forward, but society at some point in time, the United States one of them, but this is a global problem, should actually stop and think, which way do we want to go? And I think that distinction between germline and somatic is an interesting one, because from the expertise point of view, we always thought this is where we should draw a line and think it through. It's different thinking germline, somatic, and so on. From an American public views point of view, actually, our recent research showed that Americans do not care about that distinction. What they care about is the distinction between therapeutic and enhancement. So if you recall that the cover that this was showing us about, should we change the IQ or should we change, like potential Alzheimer, I don't remember what were the diseases that were on that graph, changing the, let's say, the blindness and so on, these kind of things, or the IQ may be better construed. That's actually, considered to be an enhancement approach, versus therapeutic approach would be what we're discussing with this scenario. So the report was saying, let's be careful, not assume we know what the public wants and where society wants to go.
Okay, thank you very much. What the scientific community is trying to get ahead of, I think, and this was very evident when I was in Boston and San Francisco at these other CRISPR meetings, is they want to get the community, the citizens, society involved in the conversation, get them involved early, truly understand the facts, make sure that the benefits and the risk, and understand that equation. So it's great to hear that. I think we're going to go to some of our questions here that are coming in. We have 25 upvotes on that first one. And so I can see gene editing being so expensive that it's only accessible to the wealthy. What steps should we take to limit socioeconomic gaps in access? So it's kind of interesting. That's our separate second topic. But why don't we just talk about it right now? Maybe I'll just have one comment on that, and then we can maybe go over to Jill and Dominique and Aron if you have a question about that. One of the things that we're doing as a manufacturer of Cas9 at the GMP level for clinical trials is that we've produced a batch for one particular client, but we also made more material than we needed for that particular client. So we spread the cost of GMP manufacturing over a bigger batch so we could sell smaller vials. Those smaller vials create a much lower cost point for people to buy Cas9 at GMP for clinical trials. And that's one example of how we're going to try to make it as cost-effective as possible for people to do gene editing. You saw earlier with Chris's talk about Moore's Law and sequencing and the $1,000 genome. Similar things like that are going to happen with the cost of this eventually. In the beginning, it's going to be expensive, and then it's going to go down. But to answer the question, Jill, about this socioeconomic gap between people who have access and don't have access for whatever reason, maybe you can share some thoughts on that.
So first of all, I think this leads into a bigger question about healthcare within the United States. But one thing that I will point out is that you may be familiar with gene therapy, which is different from genome engineering. So with gene therapy, you're delivering genes to a cell, whereas with genome engineering, you're actually editing the cell's own genome. And currently, as far as I'm aware, all the gene therapy that's available is available through clinical trials. So even with gene therapy, which has been around for a while, it's still not something that's widely accessible, and it's very limited in terms of its scope and in terms of its accessibility. So I think that this is an important question, but I think it has two parts, one about how we deliver healthcare, and the second thing about how we deliver healthcare through clinical trials. And so I'll turn it over to the other.
What I can add that actually I was glad that question was asked by a member of the audience, because this is one of the ethical considerations that a report from the National Academy of Science and Engineering and Medicine had pointed out, that indeed we wanted to be careful if we want this technology to be used in an ethical way to make sure it doesn't increase that gap that we already potentially see as far as access to medicine. So I think it's good to hear what Tom was saying, that already there are steps to be taken to prevent going in a direction that we wouldn't want to go, basically, right?
Right.
Yeah.
Yeah, if we can't help patients, then why are we
Exactly.
in the business, right? So, Aron, I don't know if you have some comments on that.
Right. So I agree with everything said, and to come back to Jill's point about healthcare delivery, right? So a huge challenge that isn't addressed, I think, in either of our ethical scenarios is training doctors, right? How do we train medical professionals to understand this technology well enough to talk to their patients, to recognize when a patient might be a candidate for such a clinical trial? So, with any new technology, any new therapy, it's going to start off in nucleated places, academic health centers, where you have scientists and physicians who are constantly talking to each other. And so one of the big challenges we have, I think, as scientists and researchers and and industry people is getting physicians, right, to buy in to this when it's appropriate. When is it appropriate? When is it not appropriate? How do you address your patients' questions? Because more and more patients are coming to the clinic asking about it, and we need to make sure that doctors know what they're talking about and know when something is, when the patient has something there that's real or should maybe be explored, and when it's still maybe not appropriate. And so I think that's a big gap that we don't quite know how to deal with yet.
Yeah. Maybe we can get another question here. How is safety assessed prior to clinical trials? I'm not sure if any of you guys.
Yeah, so there are many different considerations. So if we come back to gene therapy, there are different ways to deliver gene therapy. There'd be different ways to deliver the CRISPR components. And so one would be through delivering the CRISPR components directly to a human through, for example, a virus that's been a virus. Another way would be to take cells out of a human and then treat them in a dish, select for the cells that you want, that have the desired change to the genome, and then deliver those back to the patient. And so both of those approaches, I believe, are being used currently for gene therapy. And so those are two different delivery mechanisms and for genome editing. And so with safety, I think that those each have their own concerns. And I think that when you can take cells out and select for the event that you want it to occur, and then put that cell back, to me, that seems like a safer route, just given the fact that when you're doing this genome editing, the cell is deciding which repair pathway to use. And so when you deliver components directly to an organism, you can get a variety of different mutations from editing the genome, or a variety of different changes. I shouldn't necessarily say mutations.
Maybe just one other comment to follow up, Jill, and then Aron, it'd be good to get your feedback. But it's good from a terminology standpoint, what is used also in industry, Jill. So what Jill was alluding to, when we remove the cells from the body, edit them, quality control, test them, and then infuse them back into the patient, it's called ex vivo gene editing, as opposed to in vivo gene editing, which was the second scenario where you may be injecting CRISPR components directly into a particular tissue type.
Yeah.
And so there are different risk profiles, as
Yes.
Jill is mentioning, with those two different therapeutic approaches.
And one recent one involved coating gold particles with the CRISPR components and injecting them into animal models, animal disease models. And there's actually some very promising reports from those recent publications. But clearly, like gold, has its own toxicity associated with it.
Yeah.
So yeah, there'll be different things to weigh.
Aron, maybe if you have a couple more comments about.
Yeah, so I just, any sort of therapy, CRISPR or gene therapy or drug, there's a lot of preclinical work that goes into this, right? So normally, almost always, we have to start with an animal model, right? So typically, we'd like to have an animal that recapitulates some aspect of the disease so that we can show the benefit of the therapy, but we don't always have that. And so often these animal models may initially be mice or rats, and then typically once some efficacy is demonstrated in that model, we have to demonstrate in a second animal model, and that's usually a larger. So that may be a pig. It could be, if appropriate, it could be even a non-human primate model. But both of those steps would have to be sort of passed, and they'd have to demonstrate safety, efficacy, before you could move into that first phase of a clinical trial, which is that phase 1, which is a very small number of human patients, and then demonstrate success, move on to phase 2, and then eventually into a larger phase 3 trial, which might involve hundreds of patients, depending on what disease you're focused on. And so there's a very rigorous process, just like drug development, that has to be gone through by all these companies that Tom mentioned earlier, and they have collaborations with basic researchers. They have their own laboratories to do some of the preclinical work.
Yeah, and so it'd be good to get your feedback also on safety, Dominique. But just to tie it back to what I mentioned, I think this is what Aron was mentioning too, is that the innovation is happening at these smaller CRISPR companies, right? And they have the scientific ideas, but they don't necessarily have all the experience in conducting the clinical trials. And so that's why you've seen these partnerships with Editas and Celgene and the rest of them. CRISPR Therapeutics is connected with Bayer and Vertex. So Dominique, around safety, do you have?
No, it's a tricky question because, actually I teach psychology of risk, and, we, as scientists, we often think of risk assessment, but as human beings, we think of safety. We want to be safe. So there are two things when we think about it. In the real world, we're never totally safe. Like total safety doesn't exist. And I think we need to keep in mind that it's the same with clinical trials and a vast majority of the type of research we do. And that's something that we have to acknowledge, and very often it's not acknowledged, I think. And I think it's a mistake not to acknowledge that there's no totally safe way to move forward in anything, really. And I think it's something we need to ponder.
Yeah. One thing just to mention as it relates to safety, we talked a little bit, and Chris talked a little bit this morning too, about off-target effects. This is the idea that besides the actual position for the targeted double-stranded cut, is that somehow the system goes awry and cuts the genome in another location. There's a huge amount of work that is going in to optimize enzymes so that doesn't happen.
Right.
There's also a large amount of work that's going into the analytics to detect if an off-target effect actually has happened, how and why, and increase the sensitivity of that. But just to add one more comment to that, one of the things that's recently come out that's really revolutionary, it's come out of MIT, is this whole idea of a base editor enzyme. So with a base editor enzyme to do gene editing, you don't actually do a double-stranded cut in the genome. You can only do a one base pair swap based on the type of deaminase, for example, that you would fuse to Cas9. But there are all kinds of innovation that's going on around the reagents to increase the safety, because everyone knows that's a really important topic, and everyone wants to be a part of a good solution, not something that doesn't go correctly. The germline, is germline editing a long-term inevitable? Is it going to happen no matter how fast or slow we move in the US?
That's a tricky one, right? I mean, particularly something that we haven't discussed, but that is discussed a lot. That was in Hanover, in Germany, discussing the security implications of CRISPR-Cas9. And like, it goes beyond what we want to do for the human good. It could be used for bad too. So there's things that we have to think about that's related to do-it-yourself, for example. Like right now you have kits for CRISPR-Cas9 that are used by young innovators in garage, which is true. Actually, I met them. And so that's happening already. And so it's interesting to see that the markets behind the technology are moving very fast. Does it mean that, the germline gene editing is a long-term inability? That sounds like a science fiction movie title. It's like, great. I wouldn't be able to say yes or no, but the way it's moving right now, it seems like it's likely, unless you disagree with me.
No, I mean, I think it's going to happen,
right?
Whether it can become a widespread technology, I think is debatable. I do germline genome engineering in my laboratory. We do it for rats. Okay,
right.
This involves getting embryos from rats, putting the CRISPR reagent in the embryo, getting the type of genetic edit that we want to happen, and then recovering the embryo into a whole animal, and hopefully we have a new disease model. We can understand something about how that gene works. That is still, while the CRISPR technology has allowed us to do that at an efficiency that makes it feasible to do in a laboratory, it would not be feasible to do this in a human reproduction center.
So I cannot do it in my garage.
Correct, right. I think there will be scientists who are exploring. I think there will be laboratories maybe elsewhere in the world who will cater to those who want to do it or can afford to do it, and it will be done. But I don't think, I don't think it's going to lead to some supreme being, and any time in the next 20 generations of humans, we'll probably have other ways of doing that. But the idea that we could somehow change the genetic makeup of the entire human population through germline engineering just is not, it's not practical to me. So I think it would be very isolated, but I think it probably will happen somewhere.
Yeah. Can I just say one thing on top of that? So how many of you know that having 6 fingers is a dominant trait?
Right.
So it's not just limited to the Princess Bride movie, but there are people who have 6 fingers. So this is a dominant trait, but yet we all probably have 5 fingers. So I think it's important to keep in mind that unless a trait is selected for or against, it won't increase in frequency in a population. Yep. So we have to keep in mind population genetics too when we think about editing the germline. Unless the trait is selected for, it won't increase in frequency. So having one person with an edited germline or one person who's the result of an edited germline doesn't mean that that trait will necessarily spread throughout the population.
Thank you. I think it's time to move to our next scenario here and get our next voting. And this ties into kind of some of our discussion earlier. And so if you have other questions that are just on your mind, please feel free to send them in also. But new technologies also always have the potential to be transformative to society. In the genomics age, we are not only changing healthcare, science, and ag, but we are in how we relate to one another. Future genome editing therapies will be expensive and high demand and in short supply, likely to be unknown long-term health risks. So we talked a little bit about the social ramifications. Oops. So we'll switch over to Slido and wait for some of the feedback there. You're gonna see some of this, what's possible in different parts of the world. This whole social justice thing is a natural evolution of what's been happening over the last couple thousand years with different countries that have more resources than other countries. And so, the hope basically is that, for example, with sickle cell anemia, which affects over 100,000 people in the US alone and 1 million people outside the US, we have several interactions with clients right now that are focused specifically on that disease. It's a monogenetic disease where some of the first CRISPR trials will likely be in sickle cell anemia. And so, the question is, if we can do it in the US and we can make it cost effective, can we fine tune it here in the US to make it more cost effective for a broader population outside the US? So the social economic thing will continue to come. So how should gene editing resources be sold and distributed? Using a criteria based on health needs, that's 58%. Using criteria based on health and social needs, accessible to all, those that can afford them, I don't know. Maybe I can open this up to Dominique.
So Tom, just before, I would like to elaborate on a point you just made, that you said, you were talking about, we've been doing that for 2,000 years, and maybe the US can lead the way and so on. I think this is different. And actually, as somebody that's been involved in the watching the discourse, what's different with this is there is a really a different way to deal with it from an international community and the international scientific community. When you think of that National Academy of Science report we were talking about, actually the Chinese National Academy of Science asked for the American Academy to meet with them in Paris with the French one to discuss those things. Then there was another meeting in Germany with bunch of academies discussing these issues, everybody thinking we need to think this through. We cannot do like we did with genetic engineering as applied to crops, because what happened with this is that some countries, went forward, others didn't for whatever reason, and then it got at the global trade level, it got into a disaster for some countries being open to the technology, not others. The one that may need it the more don't have access to it, and so on. So I think what's good, I would say, in this is everyone is trying to say, how can we make sure that we discuss it at the international level, not to reproduce the potential fiasco we've seen for other technologies. So this is a development that I haven't seen before.
Yeah, and maybe just to touch on that, at these other CRISPR events that I've been on, genetically modified organism as it relates to agriculture and as it relates to communicating to people, society, and the market is a learning opportunity on how maybe we could get better with genome editing as it relates to how we communicate what we are doing, but also this collaboration between companies, or not companies, but countries, and different policies that are set by the FDA and the EMA and the Chinese health institutes. Okay, as the technology continues to outpace laws and policies, what systems are in place on a national, global level that would keep us from eugenic society? Anybody want to tackle that light question?
So I think it comes back to what Aron was mentioning before, where there are a lot of steps before you implement a new technology to treating humans or before it gets to a human patient. And to come back to Dominique's point about, well, there are already biohackers out there in their garages working with CRISPR. I want to remind you again of the delivery, and that these components, in order for them to work in humans, have to be delivered effectively. And that is one of the current challenges when it comes to using CRISPR-Cas9 in the germline, in somatic tissue, is delivery. So I think that one of these biohackers had a very splashy, high-profile YouTube video where he took a syringe full of CRISPR components and jabbed it in his arm. And for me as a scientist, when I see that, I think, ouch, first. And then I think, why would you put yourself through that pain? Because you're unlikely, if that needle did contain CRISPR components, you're unlikely to get those into cells efficiently. And then once you get them into cells, he was trying to mutate a gene that's involved in muscle augmentation to make a stronger forearm. I don't know if he had French Open aspirations or what, but it's unlikely that all the muscles in his arm would be edited in the same way, or even edited at all. So
it's really good to point that out.
But the Eugenius question is an
Yeah.
important one. You want to
Yeah.
comment, maybe?
Well, yeah, I think it kind of comes back to, I mean, if the question is about, you know,
It's not there.
how do, you know, are we worried that eventually in the future we're going to basically create a society of people who are completely free of disease and leave everyone behind? And I think, you know, due to the population genetics, due to the fact that unless we're selecting for these things on a population level, and this is globally, any type of gene edit that would be in the germline would not be selected for. And so it would be, I don't see how that could be done, and again, on a global scale. Now, I think, you know, what scientists are focused on, you know, is, you know, we have a family here, right? We have a family that has an awful genetic disease, right? And how can we make it so that their children and their grandchildren and their great-grandchildren don't have to worry about this disease anymore? And that's where the scientists are focused, right? And so, you know, of course, there's probably those with nefarious ideas out there, but again, I think it would be so restricted to such a small number of people that I don't see how it could be deployed in that manner.
On the other hand, you know, I'm the devil advocate, as you can see this panel. I think we need to think of the nefarious scenarios. We need to think that this is possible, and so as society, you know, put barriers, whatever we can, bioethics committees, like, you know, like regulations, funding related to, and so on, like whatever, we can think of enough. We need to make sure, not just say it's not possible, it will never happen, but say, how can we make sure it never happens? I think this is an excellent question to ask, and, you know, and think that, you know, what do we do to make sure it doesn't happen? It won't happen now. We're talking about 50 years from now that we begin to have the, you know, like potential therapies that much more, you know, efficient and so on. How can we make sure it doesn't go in a direction that we cannot, that we don't want? And I think the only way we can is by putting it on the table and showing that we care and making sure that the American public and all of us, the scientific community, show that we don't want this to happen. So we have barriers that are put in place at the legal level, regulatory level, and so on.
Yeah, it's a really good point.
Yeah.
A couple other questions here, if we can. Is informed consent possible when changes are made that affect future generations? Does anybody have any thoughts on informed consent? I'm assuming that this is at the hospital.
There is, I mean, did somebody read that report? Because apart, all those questions are related to the report. Something that was mentioned is like, let's assume you have a germline, you know, modification and so on that will affect future generations. You're not sure 100% that there won't be other mutations, something else that happens that you cannot control. You will have to follow that baby from age zero to whatever. At 18-year-old, that kid may say, you know what? I don't want to be followed. I don't want to do whatever you tell me. How do you deal with that? And this is a question that's serious, that needs to be thought through, and there's no answer yet, but it's something that needs to be pondered. And I don't know if you have any thought on that as well.
Yeah.
I don't have anything really more to add.
Yeah. So it is a question.
Oh, it's absolutely. It's one that we don't have to worry about
Yet.
engineering animal models. But yeah, I mean, that is, it's one of the many.
But you know what, that's not new when you think about it, going totally away from gene editing. Anybody that puts a picture of their baby on Facebook, you know, for all everybody to see, we never asked that baby if they wanted the picture to be on the digital world. There hasn't been a consent.
Well, also, you know, we in vitro fertilization has been
right.
going on and you know, we wanted to follow up, you know, those babies who were, you know, producing in vitro fertilization. They didn't ask to be produced that way.
Right.
And, you know, at what point do they say, hey, leave me alone?
Yeah.
Like, you know, so I think this is a question that's been around for a long time. It's not new to gene editing. It's a question that's just becoming more mainstream, I think, and a lot more people are thinking about it now.
Yeah, and I just wanted to mention, when you're like at the forefront of a new technology platform, there's going to be more questions, and there's going to be a certain level of risk that we have to absorb, basically. And getting back to patients, right, and people that are really sick and who are going to phase 1 clinical trials, and they may not have another option. It's a survival situation for them. I don't know how many of you know about the story of Emily Whitehead. She was the first patient that was treated using CAR-T therapy in Philadelphia in 2012. And they edited her T cells and they infused her T cells back into her body. She was going to die basically if there wasn't some type of intervention. So I'm sure, her parents signed a consent form, did that therapy, and today she is disease-free 6 or 7 years later, and she's actually coming to Aldevron to give a talk, because Aldevron was involved in that project in the early on days of 2012. So to advance science, we're going to have to take some risks. They're going to have to be measured, but those are a couple of good examples. Is there confidence that the trials in China are being conducted with similar regulatory oversight, regulatory standards, and peer-reviewed publications? I'm just going to say one thing about that. I'm certainly not an expert on Chinese medicine, but if you go to clinicaltrials.gov and that is the global place that people go to find out what clinical trials are going on in the world. And that's what the data that I mentioned in 2017, there are 13 clinical trials that are being conducted around CRISPR, and 11 of them are in China. Almost all the ones that are being conducted in China are around T cells, right? And T cells are somatic cells, and most likely the application is to cure cancer in the Chinese population or a specific type of cancer. So a couple other things about, let's call it universal approach or regulatory impact, is that it's probably not by accident that CRISPR Therapeutics is conducting their first clinical trial in Germany, right? So this is the first, biotech company that's in the U.S. based in Massachusetts is conducting this in Berlin for beta thalassemia. Maybe it has to do with other variables, but this is going to start shortly, so it'll be interesting to see how that plays out. But do you guys have any other comments in regards to kind of the universal approach and how things are regulated?
I would say that as a scientist, when you can repeat something that someone else has done, then it becomes more accepted. So I think that with any technology, with any results that you get in science, being able to repeat the result is, no matter where you're publishing, is the standard to meet.
Yeah, and I would say that this is really where the publication process helps us too, right? So journals have standards as far as how clinical trials and these things have to be done as well. So they're gonna, if a lab in China is developing a new therapy and they want to publish that this is a way of treating lung cancer or something like that, they're going to have to meet some criteria for that journal to understand that that the patients were given informed consent and all of these different things. So we're fortunate here in the United States to be some of the drivers of the top journals that these types of studies would be published in. But so that's part of that peer review and checks and balances comes at that level as well. But it is obviously a concern, not just with China, but with any country, right? So how do we know that any country holds themselves to the same standards that we hold ourselves to, and they're looking at us often the same way. And so I think it is an open question and part of this larger debate.
We have about 4 minutes left, so keep your questions coming. If genes are edited in diseases are slowly eradicated or looked upon with a stigma, what happens to patients who are afflicted but do not wish for treatment?
I think that this has come up with the deaf community, for example, and whether something is viewed really as a disease or really viewed as a hindrance to living a full life, So I think that this might be a case-by-case basis, and I think that our society as a whole is recognizing that we are composed of different individuals, and so I would hope that we would keep moving in that direction.
Maybe just a comment on that. On one of these panels from CRISPRCon in Boston that I remember, there was a bioethicist from England that was on the panel, and this similar question came up. And she used the example of, I think, Down syndrome, which is not particularly a target for genome editing as far as being able to cure it. But it's the idea that often people with Down syndrome are very happy people, and they don't know any other type of life, and they're perfectly happy to be just who they are. And so it's maybe, Joe, a little bit what you're saying about, you know, it's a case-by-case scenario, and it's more of a perception. Back to the questions here. How do we ensure that the results of gene editing research are applied in a socially just and equitable manner? So we talked a little bit about that earlier, but
yeah, I think we have to reinforce the fact that we have to have partnership between businesses, public universities, communities for discussion to continue. I think it is like when people are left in the dark that then you know things happen in an unjust way. So I think it's important to show up at events and discuss and raise issues and make sure that people demand answers when they want. There's nothing wrong about being concerned about things. I think it's very healthy and human, and you know, and it's the role of the scientific community to hear those concerns. So I think by continuing public governance and the public participation, that's the only way to move forward.
right. And I think you know, a lot of this research is publicly funded, right,
Yeah.
through the NIH. So the data that's generated by those tax dollars, you know, and as Aron alluded to, we thankfully are in a society where data that almost all the time that's peer reviewed, there's a process in place so that what's generated people can really believe and repeat. And so that's tied to the funding mechanism also. Any other comments? We'll move on to the next question. In the spirit of science, what is one exciting insight or discovery yielded from CRISPR-based research? Example, DNA repair mechanism. There's a lot of exciting things.
Well, I mean, I think you could start with the fundamental discovery of the CRISPR-Cas system, right? This tool, this powerful tool, has been living right under our noses. Our bodies are filled with CRISPR, right? So what is it that your body has 100 billion cells and 10 billion of those are yours? Or maybe it's trillion, I forget how that works. The other are bacteria that we live with, right? Chris talked about, Dr. Mason talked about this, right? That's part of our being, right? Those are chock full of CRISPR. These tools have been sitting there doing their thing inside, engineering genomes, changing genomes, right inside of us. And the fact that we just literally stumbled onto the fact that this technology could be applied and used in human cells and basically any DNA organism, or even RNA organism, it never stops blowing my mind that it's been there the whole time.
We're just about out of time. Dominique, Jill, Aron, thank you very much. Maybe you can any final closing thoughts before we finally wrap up.
I think it comes back to having this technology applied to making models of disease and using it within systems to understand how our neurons work, how the cells in our body work.
I think this is a great way to end this panel. Thank you so much for having us.
Yeah. So I encourage you to get involved, do research, and learn more about this exciting new technology and kind of be a part of the path forward. So thank you very much.
where's that? All right, we're just trying to get all the furniture off the stage here. So thank you so much to our keynote session on exploring the impact of gene editing. That was great, watching the polling come in. Sorry, I was looking at the screen in back and seeing all of the questions. There were tons of them. I'm sorry we couldn't get through all of them. But thank you for participating in that. Again, why we're all here is stories like we have coming up with Lauren Nedenstrom. We're going to end our program with a cancer survivor story. Certainly our industry is not only about jobs and economic output, but more importantly, it's about saving lives. Lauren Nedenstrom not only works at UW Carbone Cancer Center, but she has seen the other side of this outstanding cancer center as a patient battling thyroid cancer. Thank you, Lauren, for concluding our day and sharing your journey.
Hi, everyone. I'd like to start by thanking Lisa for inviting me to speak at the Wisconsin Biohealth Summit today. I'd like to thank all the other speakers for their wonderful talks today and for setting me up very well for this. And I'd also like to thank each and every one of you for helping me during my cancer journey. The myriad parts of the biohealth industry are responsible for me being able to stand here in this room today and talk to you. Wisconsin's state motto is forward, born out of new statehood, political discourse, and a desire to be better. This motto has served as an important reminder to me during this journey. Cancer has touched my family for decades. Both of my maternal grandparents had cancer. My paternal grandfather had cancer, and my mother is a 2-time cancer survivor. I've known since I was in high school that I wanted to do cancer research, ever since I first heard about the Human Genome Project. I had wonderfully encouraging professors in college who taught me the basic principles of good science and helped move my desire to do research forward. When it was time to apply to grad school, the first sentence of my personal statement was, "I love DNA," and I still do. I joined the University of Wisconsin-Madison Cancer Biology graduate program in the fall of 2004 in the laboratory of Dr. Dan Loeb, studying the replication of hepatitis B viruses. I was finally in my element, doing the kind of scientific research I had only read about until that point. I learned so much so quickly. The courses were stimulating, and Dan transformed me from someone who knew how to do science into a critical thinker and a good scientist. In the fall of 2005, I had the opportunity to attend the International HBV meeting in Heidelberg, Germany. I met leaders in the field, and I felt like I was taking another step forward. I was excited for the rest of my graduate career. After the meeting, I explored Spain with one of my lab mates. We walked all over Seville and Madrid, ate great food, took in the culture. And I developed a rash while I was there, and when I got home, I saw the dermatologist. That appointment was December 6th, 2005. And I had no idea how much my life was about to change. The dermatologist told me we'd probably never know what caused the rash, but that it would go away in time, and asked if he could check me for skin cancer since I had such fair skin. I mean, I'm nearly translucent, so I said sure. He looked at my back and arms and prodded around in my neck. It was the most thorough thyroid exam I had ever had. He said, "You know, your thyroid is kind of easy to feel. You should have that checked out at your next physical." So I did what any rational person would do. I freaked out a little bit. I talked to my family, and I took the next step forward and followed up with my physical, even though what I really wanted to do was bury my head in the sand and ignore that this had ever happened. But I went in, I had another thorough thyroid exam. He was pushing so hard on my windpipe, it felt like I was choking. After about 30 seconds of that, he said, "Yeah, the dermatologist was right. I think I feel something on the left side. Let's get you set up for an ultrasound." I was concerned and nervous, naturally. He reassured me, "Don't worry, you're 23, you're female. There's no way it's cancer." Well, you know where this is going. I went in for my ultrasound shortly thereafter. It's always in a warm room with what can only be described as mood lighting. That's meant to be calming or to not blind you as you stare at the ceiling while holding rather uncomfortable positions. It is not effective. The ultrasound technician was very thorough, and I left the appointment with a picture of Bob the nodule. I had to give him a name. 1 inch in diameter, left thyroid lobe, silent but deadly. I did take the next step forward and scheduled Bob for a biopsy. I also hung his picture on my fridge at home. Bob the nodule, not the ultrasound technician, just to be clear. Bob got his biopsy on January 16th, 2006. Actually, he got 5 fine needle aspiration biopsies. It was a long and painful procedure, and I made the mistake of going to that appointment alone. Looking back, I should not have been afraid to ask for help. But I'm not sure I knew what I needed. I did need a support system. My endocrinologist reassured me that I was doing great, and the likelihood that it was cancer was very low. But he kept going back for more and more samples, so what was I supposed to think? I was nervous, and I was worried, and I was scared. The very next day, I was happily sciencing along in the lab, and my cell phone rang. I stepped into the hallway and answered it. "Hello, Lauren, this is your doctor. We have the biopsy results. It looks like it's cancer." I'm sure he said more than that, but I honestly can't remember what it was. I'm pretty sure the words "good cancer" were said at some point, as if that's a thing. I tried to write down the phone number of a surgeon so I could set up a consult, but I couldn't. My doctor could tell I was struggling, so he had the surgeon call me. I know I called my parents. I called my sister too, and I don't remember much else. Moving forward had been a slow walk at this point. When you're 23, school, appointments, life, they all move without real urgency. It really seems like you have all the time in the world to get things done and to take that next step. But when the word cancer enters into the conversation, moving forward becomes an all-out sprint to get that cancer out of your body as quickly as possible. Ready or not, you're running. The next day I saw my surgeon for the first time. He explained to me that the surgery was very straightforward and we had to do it on the 24th. I hesitated and he looked at me like I had 2 heads. I said I just wanted to make sure my family could be there. I needed them to be there. It seemed like he didn't understand that. And in that moment I felt like I was nothing more than my tumor. My identity as a fully functioning human being with feelings and emotions was erased. I needed my support system. 6 days later, I was getting wheeled into the operating room, and the nurse asked what I did for a living. I told her I was studying cancer biology. "How ironic," she said. Post-op, I was told that Bob the nodule was papillary thyroid cancer, and that they had found 3 positive lymph nodes as well, bonus. The next steps had already been explained to me. I would be put on replacement hormone for a short time while I was recovering, then I would go off my medication to prepare for the dose of radioactive iodine. Any remaining cancer would absorb the radioactive iodine, my body's immune system would destroy the cells, and the cancer would be gone, and I could go on with my life as though nothing had happened. Right. During discussions with my doctors, I was told the following things, some more than once. 1, you have the good cancer. 2, you won't even notice your thyroid's gone. 3, everything will be back to normal once you're on the thyroid hormones. Well, none of those were true. 1, there is no good cancer. 2, I notice my thyroid's gone every minute of every day. 3, everything has changed. You know, interestingly, one thing they never said was, you're about to swallow a pill that contains roughly 100 millicuries of radioactive iodine, most of which your body is going to excrete as waste in an area that's really close to your reproductive organs. You're 23 years old. Have you considered freezing some eggs in case you want to start a family later? Even when I felt like a patient who knew too much due to my background in science, I was still moving forward so quickly and so scared for my life. It was impossible to come up with all the right questions to ask. So I went back to work too soon. I should have let myself feel longer, but I was bored and I missed my science. I worked as much as I could, but being off my medication in preparation for the radioactive iodine treatment was akin to slowly going into hibernation. A week before the dose was scheduled, I went in to have my blood drawn to see if my thyroid-stimulating hormone, or TSH level, was high enough that the dose of radioactive iodine they were going to give me would have a good chance of working. Now, in healthy adults, this number ranges from 1 to 5. 1 to 5. Mine was 146. 146. I was ready for the treatment. I had my dose of radioactive iodine on March 16th, 2006. It was 96 millicuries of I-131. I had to stay in isolation for approximately 1 week at home because I was radioactive. I had to use disposable plates, cups, and silverware because I was radioactive. I had to use a separate bathroom. I couldn't be in the same room as my roommates or my cats because I was radioactive. I had to do my laundry separately because it too was radioactive. I lost my sense of taste for about a month. I kept feeling worse and worse as my body caught up to the fact that I had been without thyroid hormone for so long. I was cold all the time, my hair was falling out, I was sleeping 20 hours a day. It was not fun. So moving forward took on a new meaning again. I went from a sprint to a crawl. I was started on replacement hormone right after the radioactive iodine dose in March, but it takes 6 to 8 weeks for those hormone levels to stabilize and to feel the effects, to feel better, or not. We had made an educated guess about the starting dose, waited 6 weeks, and rechecked my TSH, and adjusted the dose. Wait, check, adjust, repeat. So by May, my TSH was down to 20. By September, it was 1.73. It had taken me 6 months to start to feel human again. Now, the hardest part of this 6 months period was not being able to do my job well. My brain was so foggy and I couldn't get my hands to do what my head wanted them to do. I ended up graduating in December of 2006 with my master's degree in cancer biology and a lot of first-hand experience to go along with it. I got a job working in the laboratory of Dr. Christine Ceruggi, studying the role of regulatory T cells in pediatric allergy, asthma, and autoimmune diseases. In her lab, I was introduced to the University of Wisconsin-Madison flow cytometry core facility, where I'm currently employed, and the field of flow cytometry. While absorbing all the information and exciting science at my new job, I was still plodding forward through the never-ending follow-up and monitoring appointments. Blood tests, check TSH, followed by scans and long discussion with the endocrinologist to try to make sense of all of the side effects they had assured me would not be a problem. My constant companions were weight gain, chronic pain, difficulty regulating body temperature, brain fog, and heart palpitations due to the dose of thyroid hormone required to keep the cancer from coming back. I was not who I was before this started. In 2010, my endocrinologist called me to tell me that some of the lymph nodes in my neck had grown over the previous 6 months. He wanted me to have another low-dose radioactive iodine scan to see if there was any uptake. It was negative, but my blood test showed my tumor marker was a little bit elevated. I dubbed this ninja cancer. Something was there, but we couldn't see it yet. I felt like I was running again, but in circles this time. We watched and we waited. My anxiety was at an all-time high. I was depressed as well, surprise, surprise. So I started seeing a therapist to figure out how to handle this new situation. I learned that it was okay and even necessary to mourn both the life I had before my diagnosis and the life that I thought I was going to have in the future. I learned that taking care of myself was important and was in no way selfish. I changed my diet. I tried yoga. Once I saw that I was capable of making those adjustments, it was easier to move towards a new normal. And for the first time, I felt like I could influence what that new normal could become. In June of 2012, after another whole body scan, I got another phone call at work while I was happily sciencing along. The cancer was back. I remember shaking and crying and being angry. It took a long time for me to compose myself before I was able to go home. I immediately reached out to my support system, and wow, did they reach back. I would not have gotten through a second round of all of this without them. I continued therapy, I learned about mindfulness, I learned to stop the anxiety when it would creep up on me unexpectedly, I learned how to deal with PTSD. My sister and I were living together, and she made sure I was happy and loved throughout that recurrence. And she even introduced me to my husband. It's been 6 years since my recurrence, and the meaning of moving forward is going to continue to change for the rest of my life. What I know now is that every step, big or small, must be deliberate. Every step, big or small, must be appreciated. Every step, big or small, is important. Please remember that. Without the steps each of you have taken, many cancer survivors, myself included, would not be here today. I find myself on a different path today than I could have imagined. It's been winding and bumpy and scary and surreal, but I'm still moving forward. I'm so happy with the person I've become through all of this, and I wouldn't change a thing. Thank you.
I say you take it out. Thank you. Lovely. That was awesome. So thank you, Aron. Thank you for sharing that story. That's not easy. And it's certainly not easy what you went through. Thank you to all of you that are impacting lives like Lauren. And thank you to our medical profession, the researchers. Again, this is what we live for. So that concludes the programming portion of the 2018 Wisconsin Biohealth Summit. Again, thank you again to all the participants, to Dr. Chris Mason, again for all of our sponsors, for supporting this conference, for all of you for attending. I do want to remind you, and there's our sponsors. There we go, career sponsors. Okay, so for the career fair, this is new this year. That is next to our reception. Again, this is the second floor. And please, students that are out there, go check it out, and any others. We got a lot of great companies there to meet with you. And then at our reception, we thank CRB. This is a great networking opportunity, and thank you very much to CRB for sponsoring the reception this year. So before I let you go, you know I cannot end a day without playing Pink. So final words, I'm going to say it one more time. Please go forth with energy, find ways to collaborate, have your voices heard.